Guys, good day, praise be Jesus and Mary. How are you? I hope you are fine today. So I am here to discuss about the chapter 11, which is the membrane transport of small molecules and the electrical properties of membrane. So this is an image of a plasma membrane, a lipid bilayer. So this plasma membrane or the cell membrane, it provides the protection for a cell. Then it also provides a fixed environment inside the cell. So it has several functions. So the plasma membrane has several functions, and one of this is to transport the nutrients inside the cell and for the toxic substances out of the cell. So the membrane transport proteins. These membrane transport proteins helps attain the goal of a cell to penetrate nutrients and other materials in hydrophobic membranes. So, for the membrane transport proteins, there are two classes. These are the transporters and the channels. So, for transporters, it undergoes sequential conformational changes to transport a specific small molecule across membranes. And channels, they form narrow pores, allowing positive transmembrane movement. So we will know more about these two classes in the next slides of this chapter. So we'll also know about the principles of membrane transport, the transporters and active membrane transport, the channels and the electrical properties of membranes. So for number one, the principles of membrane transport. So what are the principles of membrane transport? First is the protein-free lipid bilayers are impermeable to ions. So, since there are also nutrients or molecules that can be penetrate to the lipid bilayers, and these are the hydrophobic molecules. These are soluble in oil. These are also a polar molecules, and and they diffuse. They have a faster rate of diffusion. While for small uncharged polar molecules, they diffuse across bilayer also, but slower than the hydrophobic molecules. While the large uncharged polar molecules, they diffuses but much more slowly than the small uncharged polar molecules. So diba since slow na gani, so mas slower ang small uncharged polar molecules than the hydrophobic molecules, which have a faster rate of diffusion. So ang large uncharged polar molecules is slow, gamay ra gid kayo ihang rate for diffusion than the two. So for ions, these are the charged molecules. They are impermeable to the lipid bilayers. So in this image, we can see that the hydrophobic molecules like the oxygen molecule, the carbon dioxide, the nitrogen molecule, and the steroid hormones have faster rate of diffusion of the lipid bilayer. While for small uncharged polar molecules like the water, the urea, the glycerol, they diffuses but much more slowly. But much more slowly than the hydrophobic molecules. While for the large uncharged polar molecules like the glucose and sucrose, they can diffuse but they are much more slower than the small uncharged polar molecules. So see for the yellow one, mas tin iyahang rate for the diffusion to for the diffusion of the lipid bilayers, while for ions like the hydrogen ions, the sodium ions, the potassium ions, the calcium ions, they cannot penetrate to the lipid bilayer. So how can they penetrate to our to to the lipid bilayers? We will know the process on the later slides. So we really need these nutrients in our body. So we will know on how they will penetrate the lipid bilayers despite being impermeable to it. Second principle of the membrane transport. There are two main classes of membrane transport proteins. These are the transporters and the channels. As I have said earlier, so for transporters, it is also called as the carriers or the permeases. So they bind the specific solute to be transported and undergo a series of changes that alternately expose solute binding sites on the one side of the membrane and then on the other to transfer the solute across it. While the channels, they don't bind the specific solutes. They don't bind the solute. Instead, they interact with it. They interact with the solute to be transported much more weakly, and they form continuous pores that extend across the lipid bilayer. So the third principle of membrane transport is that active transport is mediated by transporters coupled to an energy source. So before explaining that, we should also know that there are two types of transport. These are the passive transport and the active transport. So what is this passive transport? So this passive transport is a process where all the channels and many transporters allow solutes to cross the membrane only passively. So meaning passively, it is a downward motion, a downhill motion. While for the active transport, it is mediated by transporters whose pumping activity is directional because it is tightly coupled to a metabolic source of energy. So always remember that active transport always coupled to a metabolic source of energy kaya ang iyahang way of transport is pa-uphill. So upward iyahang motion sa iyahang transport. So since para ma-attain ang iyang upward motion, we need that metabolic source of energy. So for concentration gradient, this concentration gradient is the difference in the concentration and the size of membrane that drives the passive transport and determines its direction. So the difference, ang concentration gradient is that from higher concentration to low concentration. So as we can see that pa downward ang ihang motion sa ihang transport. So ang ihang transport is from higher concentration of solutes to the low concentration of solute. So and then the membrane potential. So what is this membrane potential? This is the difference between the concentration gradient and the electrical potential difference or the EPD. So this electrical potential difference is that it is also known as the voltage. It is an external work needed to bring a charge from one location to another location. While the electrochemical gradient, it is a net driving force by combining concentration gradient and electrical gradient. So, for after we have learned the three principles of membrane transport, we then proceed to the transporters and the active membrane transport. So, for cells to carry out active transport in three main ways. So, cells carry out active transport in three main ways. These are the coupled transporter, the ATP-driven pump, the light-driven pump. So, for coupled transporters, it harnessed the energy stored in concentration gradients to couple the uphill transport of one solute across the membrane to downhill transport of another. So, coupled transport, for muna siya ang orange is our solute. Then, ang energy is the red. So, it couples the uphill transport of the solute across the membrane to the downhill transport of another, which is the energy. For the ATP-driven pumps, it coupled the uphill transport to the hydrolysis of the ATP. And third way is that light or redox-driven pumps, it coupled the uphill transport to an input of energy from light as with bacteriorhodopsin or from redox reaction as with the cytochrome C oxidase. So it is this light or redox driven pumps also known in this way is known in bacteria, the archaea, the mitochondria, and the chloroplast. Another is that the transporter is shown in three conformational states. So, the three conformational states are the outward pen open, the occluded, and the inward open. So, for outward open state, the binding site of solutes are exposed on the outside. So, ang iyang binding site, exposed siya outside. So, while the occluded state, same sites are not accessible from either side. So, ang iyang solute is nara sa center. While the inward open state, sites are exposed in the insides. So, ang iyang site, exposed na siya inside. So, there are 6 things that we will discuss about the transporters and active membrane transport. So, number 1 is that active transport can be driven by ion concentration gradient. Number 2, the transporters in the plasma membrane regulate the systolic pH. 3 is that an asymmetric distribution of transporters in epithelial cells underlies the transcellular transport of solutes. Number 4 is the P-type ATPase pumps of the calcium ion into the sarcoplasmic reticulum in the muscle cells. And number 5, the plasma membrane of the sodium ion, potassium ion pump establishes the sodium ion and the potassium gradient, while the ABC transporters constitute the largest family of membrane transport proteins. So for number 1, that active transport can be driven by ion concentration gradient. So there are 3 transporters act as these 3 types. So for uniporters, as uniport, the symport, and the antiport. So for uniporters, it is where the transporter that passively mediate the movement of a single solute from one side to the other. So uniport, pa-downhill ang iyahang, pa-downward ang iyahang motion. So it transport passively, it mediates the movement of single solute from one side to another. While the symporters are also called as the co-transporters. They perform the simultaneous transfer of a second solute in the same direction. So, same direction. Iyahang ang solute and the co-transported ion, ang transported molecule and the co-transported ion, same direction ang iyahang transfer to the other direction. So for antiporters, or also known as the exchangers, they perform the transfer of a second solute in the opposite direction. So, kabaliktaran siya sa atong same porters. Ang atong exchangers or the antiporters perform the second solute in the opposite direction. So, that. So this image is the mechanism of glucose transport fueled by the sodium ion gradient. That's that. So for this figure, it is the transporter alternates between inward open and outward open states via an occluded intermediate state. So binding of the sodium ion and the glucose is cooperative, so that is. The binding of either solute increases the protein's affinity for the other, and since the sodium ion concentration is much higher in the extracellular space than in the cytosol, glucose is more likely to bind to the transporter in the inward facing state. So this. So diba, from first, it is occluded empty. So ang glucose is the blue, and then the ion, the sodium ion, is represented by the red circle. So from the occluded empty, since wala kaayo sodium ions, since wala kaayo ay ang concentration gradients, electrochemical gradient for the sodium ion sa atong cytosol is much lower. So from occluded empty, so wala siya sa middle, to the outward open. So then the sodium ion enters the center to the occluded occupied. So since from the occluded empty to the occluded occupied, then to the inward open. So pag inward open, na-transport ang iyahang mga ang sodium ions and the glucose. So from the occluded empty, ni-open siya sa outward open. Then again, from occluded occupied, since nakakuha na siya sa mga ions nga iyahang need. And then, for the inward open, para ma-release niya downward for the ions and the glucose. So, another process is that occluded empty, since the ions and the glucose is already transported to the other side. So the glucose concentration gradient now, ang iyahang concentration gradient is for cytosol, mas daghan na siya ang atoang glucose content than the outside. So for number 2, the transporters in the plasma membrane regulate the cytosolic pH. So for lysosomal enzymes, so also, the transporters also functions as enzymes. So, for lysosomal enzymes, they function best at low pH. So, that equals to the pH of 5. While the cytosolic enzymes function best at close to neutral pH, which is equal to 7.2. But how? How can they regulate the how can the plasma membrane regulate the cytosolic pH? So, the hydrogen ion is directly through the hydrogen ion is directly transported out of the cell, or the bicarbonate is brought into, or the carbonates rather, is brought into the cell to neutralize the hydrogen ion into the cytosol. So according to the reaction, so our carbonates as a plus the hydrogen ion, the react then the reactants will be the water and the carbon dioxide. Also, the other mechanism is that when the hydrogen ion pumps maintain the low pH in lysosomes as well in endosomes and secretory vesicles. So this hydrogen ions pumps use the energy of ATP hydrolysis to pump the hydrogen ion into these organelles from the cytosol. So that's how the plasma membrane regulate the cytosolic pH, which function best at close to neutral pH equals to 7.2. So for number 3, an asymmetric distribution of transporters in epithelial cells underlies the transcellular transport of the solutes. So there are three principal classes of ATP-driven pumps. So number 1 is the principal classes, and number 1 is the P-type pumps. It is called P-type because they phosphorylate themselves during the pumping cycle. So this class includes many of the ion pumps that are responsible for setting up and maintaining gradient of the sodium ion, the potassium ion, the hydrogen ion, and the calcium ion across cell membranes. So P-type because they phosphorylate themselves during the pumping cycle. So for ABC transporter or the ATP binding cassette transporters, they differ structurally from P-type ATPases and primarily pump small molecules across the cell membranes. So for B-types, pumps, it is a turbine-like protein machines constructed from multiple different subunits. So you might be wondering why I have said earlier that there are three principal classes of ATP-driven pumps. Well, I wrote four. So for F-type ATPases, it is a distant family of the B-type pumps. So for F-type ATPases, it is more commonly called as the ATP synthesis because they normally work in reverse. So instead of using the ATP hydrolysis to drive the hydrogen ion transport, they use the hydrogen ion gradient across the membrane to drive the synthesis of ATP from ADP and phosphate. So this is the image of the three types of ATP driven pumps. The P type. The three main classes are the P-type pump, the ABC transporter, the B-type proton pump, and the distant family of the B-type proton pump is the F-type ATP synthase, which work in reverse. ATPase pumps the calcium ion into the sarcoplasmic reticulum in muscle cells. So the calcium ion pump or the calcium ion ATPase in the sarcoplasmic reticulum membrane of a skeletal muscle cells is a well understood P-type transport ATPase. So as we have known that the P-type pumps is called because they phosphorylate themselves during the pumping cycle. So this sarcoplasmic reticulum is a specialized type of endoplasmic reticulum that forms a network of tubular sacs in the muscle cell cytoplasm, and it serves as an intracellular store of the calcium. So when an action potential depolarizes the muscle cell plasma membrane, the calcium is released into the cytosol from the sarcoplasmic reticulum to the calcium release channels, so stimulating the muscle to contract. So the calcium ion pump, which accounts for about 90% of the membrane protein of the sarcoplasmic reticulum, they moves the calcium ion from the cytosol back into the sarcoplasmic reticulum. So the endoplasmic reticulum of non-muscle cells contain a similar calcium plus calcium ion pump but in smaller quantities. So, where can we find this archoplasmic reticulum? Of course, sa buol sa yaha which needs the calcium, which is the bone. So, this sarcoplasmic reticulum is from the skeletal muscle cells. This is the specialized type of the ER endoplasmic reticulum that form a network tubular sacs in the muscle cell cytoplasm. So so it also for the calcium ion release channels, it also stimulates our muscles to contract. So for number 5, the plasma membrane sodium ion potassium ion pump establishes the sodium ion and the potassium ion gradient across the plasma membrane. So the for concentration of the potassium ion is typically 10 to 30 times higher inside cells than the outside. So whereas the reverse is true of the sodium ion. So mas higher ang concentration outside sa sodium ion than the potassium ion. So reverse lang sila. So from the sodium ion potassium ion pump or the sodium ion potassium ion ATPase they are found in the plasma membrane of virtually all animal cells that maintains the concentration differences. So since the sodium ion potassium ion pump drives 3 positively charged ion out of the cell for every 2 it pumps in, it is electrogenic. So what is this electrogenic? It drives a net electric current across the membrane, so tending to create an electrical potential with the cell inside being negative relative to the outside cell. So as we have known that this image, this figure, this is the function of the sodium ion potassium ion pump. So this P-type ATPase actively pumps. It is also a P-type. This P-type ATPase actively pumps the sodium ion out of the potassium ion into a cell against their electrochemical gradient. So, it is structurally closely related to the calcium ion ATPase but differs in its selectivity for ions. So, for every molecule of ATP hydrolyzed by the pump, there are 3 sodium ion that are pumped out and 2 potassium ion. There are 2 potassium ion pumped in. So we will remember that in every molecule of ATP hydrolyzed by the pump, there are 3 sodium ion are pumped out and there are 2 potassium ion that are pumped in. So for number 6, which is the ABC transporters constitute the largest family of membrane transport proteins. So they are called the ABC transporter because each member contains 2 highly conserved ATPase domains or ATP binding cassettes on the cytosolic side of the membrane. So there are family of membrane transport proteins, and one of this is the multi-drug resistance or the MDR protein. It is also called the P-glycoprotein. It is present at elevated levels in many human cancer cells and makes the cell simultaneously resistant to a variety of chemically unrelated cytotoxic drugs that are widely used in cancer chemotherapy. Next is the cystic fibrosis transmembrane conductance regulator protein or the CFTR. It is it was discovered through studies of the common genetic diseases called the cystic fibrosis. Thus, in most vertebrate cells, an ABC transporter in the endoplasmic reticulum or the membrane or the ER membrane actively pumps a wide variety of peptides from the cytosol into the ER lumen. So for this figure, it is the small molecule transport by a typical ABC transporter. So ABC transporter consists of multiple domains. So typically, two hydrophobic domains each built of six membranes spanning alpha helices together with a translocation pathway and provide a substrate specificity. So for A, it is a bacterial ABC transporter. So they are both importing and exporting the ABC transporter are found in bacteria, and ABC importer is shown in this cartoon. So the crystal structure of the bacterial ABC transporter is shown in this figure A. So for figure B, in eukaryotes, a eukaryotic ABC transporter. So for figure A, bacteria to siya, and for letter B, it is eukaryotic ABC transporter. So in eukaryotes, most ABC transporters exports the substances. So either from the cytosol to the extracellular space or from the cytosol to a membrane-bound intracellular compartments such as the ER or endoplasmic reticulum or from the mitochondrial matrix to the cytosol. So here, from the cytosol to the intracellular compartment, from the cytosol to the intracellular compartment. While sa bacterial is kabaliktaran siya, it's from the intracellular compartment to the cytosol. So this figure is the auxiliary transport system associated with transport ATPases in bacteria with double membranes. So from outer membrane, periplasmic space to the inner plasma membrane since double siya. So sa cell exterior, si diba? So ABC transporter here in the cytosol to the periplasmic space, which forms a cycle, periplasmic substrate binding proteins with bound solute and solute-free periplasmic substrate binding the protein, so they help the protein. And then from the outer membrane, diba sa bacteria is from the intracellular compartments to the cytosol. So from the outer membrane, nisulod ang iyahang mga solute through the porins and then to the periplasmic space. So this periplasmic space, they help the substrate to bind the proteins and then down to the cytosol. Ang solute, nipadulong din sa cytosol. So that is for the bacteria with double membrane. So the third topic, which is the channels and the electrical properties of the membranes. So for this topic, we will not go in depth, and I will be just discussing the summary or some of the topics of um this. So for the channels and the electrical properties of membrane. So for ion channels from aqueous pores across the lipid bilayer and allow the inorganic ions of appropriate size and charge to cross the membrane down their electrochemical gradient at rates about 1,000 times greater than those achieved by any known transporter. So these aqueous pores, also known as the aquaporins. aquaporins, so which are permeable to the water but impermeable to the ions. So aquaporins is a water channels found in epithelial cells of the kidney or exocrine cells. So the channels are gated and usually open transiently in response to specific perturbation in the membrane, such as a change in membrane potential of the voltage gated channel for the voltage gated channels or at the binding of a neurotransmitter to the channel transmitter gated channels. So we have here the two types, which is the closed and open. These are the typical ion channel which fluctuates between closed and open conformations. So the ion channel shown here in cross section forms a pore across the lipid bilayer only in the open conformational state. So open conformational state. So the pore narrows to atomic dimension in one region where the ion selectivity of the channel is largely determined and another region of the channel forms the gate. So for the closed conformation, we have here this is the gating of the ion channel. So this schematic drawing shows the several kinds of stimuli that opens ion channels, and mechanically gated channels often have cytoplasmic extensions that link the channel to the cytoskeleton. So they link the channel to the cytoskeleton from the closed conformation and the open conformation. So voltage gated here sa green, they're represented by the color green. For the ligand-gated extracellular ligand, represented by the color blue, iyang pa-open is the pa-outward iyang pag-open. While for the ligand-gated intracellular, of course, intra, so pa-inside iyang opening. Well, for the mechanically gated, represented by the color red. So, these are the schematic drawing that shows the several stimuli that open the ion channel or the gating of the ion channels. So, the potassium ion selective leak channels have an important role in determining the resting membrane potential across the plasma membrane in most animal cells. And the voltage-gated cation channels are responsible for the amplifications and propagation of actions potential in electrically excitable cells and such as neuron and skeletal muscle cells. And transmitter-gated ion channels convert chemical signals to electrical signals at chemical synapses, and excitatory neurotransmitters such as the acetylcholine and glutamate. So, it is the open transmitter-gated cation channels and thereby depolarize the postsynaptic membrane toward the threshold level for firing and action potential. So, inhibitory neurotransmitters such as the GABA and lysine open the transmitter-gated, the chlorine ion and the potassium ion channels and thereby suppress firing by keeping the postsynaptic membrane polarized. So for ion channels to work together in complex ways to control the behavior of electrically excitable cells. So a typical neuron, for example, receives thousands of excitatory and inhibitory inputs, which combines by spatial and temporal summation to produce a combined post-synaptic potential or the PSP at the initial segment of its axons. For better understanding about these channels, I will show you a 3-minute video that talks about not only the channels but also the membrane transport of small molecules.
All animal cells are enclosed in a plasma membrane, which consists of two layers of phospholipids. The hydrophobic nature of the cell membrane makes it intrinsically permeable to small nonpolar and uncharged polar molecules, but non-permeable to large polar molecules and charged particles. Charged particles, such as ions, must use special channels to move through the membrane. Transport of a molecule can be passive or active. Passive transport does not require energy input because it moves the molecules downhill, for example, from higher to lower concentration. Active transport, on the other hand, moves the molecules against their gradients and therefore requires energy expenditure. Ion channels permit passive transport of ions. These are transmembrane proteins that form pores for ions to pass through. Most ion channels are specific for a certain type of ion. Ion channels can be classified by how they change their open-closed state in response to different factors of the environment. Common types of ion channels include leak channels. These channels are almost always open, allowing more or less steady flow of ions. Examples are potassium and sodium leak channels in neurons. Ligand-gated ion channels. These channels open upon binding of a ligand. They are most commonly found at synapses, where neurons communicate via chemical messages, or neurotransmitters. An example is the GABA receptor, a chloride channel located on postsynaptic neurons. It opens upon binding to GABA, a neurotransmitter released by the presynaptic neuron, and allows chloride ions to flow into the cell. Voltage-gated ion channels. These channels are regulated by membrane voltage. They open at some values of the membrane potential and close at others. These are the channels that underlie action potentials in neurons and cardiac muscles.
So that's all for chapter 11, and thank you for watching, and I hope you learned something from it.